So the first block of slides talked about carbon metabolism through some of the most common metabolic pathways that microbes use. This group of slides is looking more at nitrogen metabolism, specifically protein and amino acid catabolism. And so the most basic enzyme required to eat proteins is a protease that hydrolyzes protein to amino acids by breaking up the peptide bonds that link amino acids together in polypeptides. Deamination is typically required because that's the removal of an amino group, sort of the dismantling of the amino acids into individual components, resulting in organic acids that can be readily converted to pyruvate or the 2-carbon acetyl-CoA or other TCA cycle intermediates. And these can then be fed into the TCA cycle and oxidized to produce electron carriers such as FADH2 and NADH, but also to produce energy. And again, the carbon skeletons can be used for biosynthesis. And one of the main processes here is something called transamination, which I'll talk about. So the proteases typically hydrolyze peptide bonds, and as you can see here, with the addition of water, the protease can attack this bond here, breaking that into individual smaller units, either individual amino acids in peptides or two peptides and so forth, as you see here. So the nomenclature for proteases or peptidases, as they're referred to interchangeably, is indicated here. So an endopeptidase or an endoprotease is a hydrolytic enzyme that breaks peptide bonds that are internal to the N-terminus and C-terminus, so somewhere inside here. And so again, these guys typically will break these bonds internally, resulting in the formation of two smaller peptides, polypeptides. An exopeptidase, on the other hand, can be broken into two categories, either an aminopeptidase that breaks the immediate peptide bond to the to the proximal amino acid, and the immediate peptide bond that's just next to the terminal amino acid. So a carboxypeptidase cuts at the peptide bond at the carboxy terminus, and an aminopeptidase cuts at the peptide bond at the amino terminus. And so in the lab, we'll be working with, down the road, we'll be working with an aminopeptidase. This next slide just kind of overviews some of the transporters, permeases, and so forth that microbes might use to transport these things once they break them down. There's a couple variations on the theme here. So number one, you see a cell wall associated protease that's sticking out into the world and can chop up polypeptides. This is PRTP. And again, this is for starter culture type bacteria like Lactococcus, which we might use to make cheese, but by no means restricted to these types of organisms. These sophisticated types of amino acid and polypeptide uptake systems occur in lots of microbes, but it just so happens that that starter culture bacteria are pretty prolific amino acid utilizers. And one of the reasons for that is milk proteins are obviously quite abundant. And so casein is the major milk protein, and here's a representation of it as a big polypeptide. And then these cell surface associated proteases can chop that into individual oligopeptides, so smaller peptides. And then these can be transported by an oligopeptide permease. And so one thing I'll say too is that it's not always cell wall associated. Proteases can be produced in the cytoplasm and pumped out into the world as well. So it's reminiscent of what you guys did in the lab, for example, with the amylase assay, where you looked at, is the activity cell surface associated or secreted? You can do the same thing with proteases using a similar methodology, but obviously with different substrates and chemistry, but the principle is the same. So an oligopeptide permease like this operates basically as an ABC type transporter, an ATP binding cassette type transporter, and I'll explain that later. The individual amino acids can be, so these can be broken down further by secreted proteases, and these individual amino acids can be taken up by amino acid transporters. And then there's di and tripeptide transporters, as you see here. So here's a tri and a dipeptide, and here's two flavors of these types of transporters. There's DTPP, which is an ABC type transporter, an ATP binding cassette type transporter, and then DTPT, which operates on proton motive force, meaning that when the microbe is transporting electrons across its electron transport chain and pumping protons into the world, the controlled reentry of those protons facilitates the co-transport of these di and tripeptides. So some of the lab-based protease detection methods, and as I said, we're going to do some of this in the lab, are indicated here. And so some of the old school stuff, like what we do in Micro 224, where we look at these, plating assays. So here's skim milk auger, and we'll put a microbe like Bacillus subtilis on there. You use Bacillus subtilis also for the amylase assay in lab, but it's because again, it's a prolific producer of secreted enzyme activities. It's a classic soil microbe that's a saprophyte that is whose job it is to clean up all the garbage. And in order to do that, the microbe has to produce a lot of extracellular enzymes. So here's the microbe heavily inoculated on the plate, and then that window around the colony illustrates where the microbe has eaten up all the opaque casein proteins to create that translucent window around itself. Another gold standard in lab-based protease detection is gelatin hydrolysis. And so you can use gelatin that's pretty solid if you tilt the tube sideways. But if you put a gelatinase producing microbe who's producing that type of protease, it'll liquefy the medium and the medium becomes runny. But we'll use one of these fluorescence type assays, which I find much more exciting. And it's actually neat to see the activity in culture, but I'd also like to see the fluorescence activity type assays. Again, it's more sensitive, and these give more versatility in terms of the types of experiments that you can do. And so here's a casein protein, and it's been labeled with these fluorescent markers. But because it's all folded up like this, the fluorescent markers are quenched. And so if the microbe produces a casein degrading enzyme, a protease essentially, that can break this polypeptide, it'll break it into pieces and release those fluorescent dyes. And therefore you put that into a fluorimeter and you can detect that. This is the actual fluorescent substrate that we will use. And so it's basically has a fluorescent marker attached to it. And so here's the peptide bond that's broken. And so what you're looking at is essentially an aminopeptidase, which you'd be looking for using this type of assay as an aminopeptidase type assay or enzyme activity. So in this particular case, here's your amino acid here. Here's your peptide bond, and that's linked to this fluorescent substrate here. And so if you have an enzyme that can break this thing, an aminopeptidase type enzyme that can break this proximal peptide, it'll release this into the medium and cause fluorescence. So again, we'll work with this. This is actually a leucine aminopeptidase. So here's the leucine amino acid peptide bonded to a fluorescent dye. And so data output could look something like this. And so on the y-axis is fluorescence units per OD. So as the culture grows, you can detect changes in activity. And then on the x-axis is time. And so what we're asking is a fundamental question: is the enzyme activity cell associated, or is it secreted? And so in this particular case, and the other question that we can ask is, can the activity be inhibited by a zinc metalloprotease inhibitor? Because a lot of these enzymes require zinc as a cofactor. And so the experiment's pretty straightforward. You grow some microbes in culture, and then over time you grab aliquots of culture and you separate it into fractions like you guys did. You spin it down and you get the cell pellet, and you keep the supernatant, you pull the supernatant off separately, and then you can test each fraction separately for enzyme activity. And so here you see the enzyme activity increasing in the cell fraction and being basically non-existent in the supernatant fraction. Similarly, you can add once you know that the activity is associated with the one fraction or the other, in this case it's the cell fraction, you can take the cell fraction and mix that with a zinc metalloprotease inhibitor, such as Zinkoff, and show that you can abolish the activity. So you get two pieces of information here. Again, that the activity is cell-associated and that the activity can be interfered with by a zinc metalloprotease inhibitor, suggesting that it does require zinc as a cofactor. So again, once the microbe has broken down polypeptides into individual amino acids, the individual amino acids have to be taken apart. And one of the processes is called deamination, where the microbes will pull off amino groups. And so one method of doing that is non-oxidative deamination, where microbes will make an enzyme like a serine dehydratase. So here's serine, and the dehydratase, as the name suggests, will pluck off water at first and produce this intermediate aminoacrylate. And then as more water is added back, the enzyme can then pull off the amino group and then produce pyruvate. And then pyruvate can be used, can be shunted into TCA cycle, for example, or it can be shunted in to make other amino acids or for other biosynthetic processes. Another type of non-oxidative deamination occurs here. You see this sulfhydrase. In this particular case, the sulfhydrase requires this pyridoxal phosphate cofactor, and the desulfhydrase will rip this sulfur group off of cysteine, because again, this is cysteine, an amino acid that contains sulfur, and then resulting in an amino acid, which then the desulfhydrase can then pluck the amino group off of. So right here, this guy gets plucked off. And again, you make pyruvic acid or pyruvate, and then this can again be shunted into TCA cycle and so forth. So this enzyme desulfhydrase or serine dehydratase, two examples of enzymes that undergo non-oxidative deamination. Oxidative deamination, on the other hand, looks like this, and again, as the name suggests, oxygen is required. And so phenylalanine deaminase is an example of an oxidative deamination enzyme. And this enzyme operates through the dihydrofolate reductase cofactor 2, enzyme rather. So you need dihydrofolate reductase in order to facilitate this reaction. So phenylalanine deaminase, dihydrofolate reductase, oxygen, water, and then in this process, basically the microbe will pluck this amino group off, as you see here, and then release that free ammonia. So hopefully in some of your classes, like Gen Micro, you did one of these phenylalanine deaminase tests, and so in that particular case, you would do the reaction, grow the microbe on phenylalanine-based medium, and then expose that medium to ferric chloride if either of these end products is present, the phenylpyruvic acid, for example, will react with the ferric chloride to produce this green end product. So again, oxidative deamination using phenylalanine deaminase as an example. And again, hopefully you did this in Micro 224. I think the way we did it in Micro 224 was with little tablets, little ferric chloride tablets, and so you add those to a liquid medium and then look for the green color formation as a result of these end products reacting with the ferric chloride. So transamination occurs through transaminase, and so when a microbe breaks down an amino acid to release these amino groups, obviously the microbe has to be able to use these amino groups to do something with them, and usually it's putting them onto carbon skeletons to make additional nitrogen-containing compounds. And so here's an example of that. But in this particular case, a transaminase enzyme is not picking up free ammonia. It's basically taking ammonia that's bound off of something that you don't need it as much of and putting it onto something else to make something new that you need more of. So in this particular case, imagine you have an abundance of alanine and you need more glutamate. And so a transaminase would pull this amino group off of alanine and stick it here onto alpha-ketoglutarate. Remember, alpha-ketoglutarate comes from the TCA cycle, so it's a common intermediate in the TCA cycle. And so the amino group gets plucked off, put here, and then you end up with pyruvate, which can go into the TCA cycle, and glutamate, which can then be used as a entryway for nitrogen into the production of additional amino acids, or the amino acid glutamate can be used in and of itself. So another example of that, and showing you more of the versatility. So imagine you've got an amino acid here. Here's just the generic amino acid structure, so it could be anything. There's a lot of different transaminases, and here, here you're looking at a very specific transaminase reaction from alanine to make glutamate. But in this particular case, there's lots of transaminases that I wanted to point out. And so the amino acid group gets, the amino group gets pulled off, stuffed onto oxaloacetate. That allows you to make, for example, in this case, aspartate. The amino group from aspartate can be plucked off and put onto alpha-ketoglutarate to make glutamate. And then obviously the glutamate can be metabolized by using glutamate dehydrogenase, for example, to pull that amino group off there and make reducing power and so forth. Another key macromolecule that has to be consumed are fatty acids. And so fatty acid metabolism is primarily driven by beta oxidation. And so again, microbes produce all kinds of lipase type enzymes that will break down lipids and allow them to perform beta oxidation. And so in this particular case, here's a fatty acid right here. And as long as there's a little bit of CoA and energy around, this acyl-CoA ligase can stick these guys on in an energy-dependent way, the CoA onto the terminal end of the fatty acid. And then an acyl-CoA dehydrogenase, as you see here, can oxidize that terminal region here, and again make reducing power in the form of FADH2 that can be sent to electron transport. And basically all you're doing is modifying the terminal region here. So here's a dehydrogenase, and again you're making more reducing power. And then at the end of all this process, the ethyolase that you see here will grab CoA and stick it on, basically cut this, and then, cut here. Well, the thiolase will cut here and stick this CoA here and give you this 2-carbon acetyl-CoA. And then this acetyl-CoA can then be metabolized to make energy reducing power and more in terms of FADH2 and NADH. So you're making reducing power. So each time you do a modification of the terminal fatty acid region, you make a ton of reducing power, and all this can dump its electrons into electron transport to make a bunch of energy. But you're also making acetyl-CoA, which directly can go into TCA cycle to make even more energy and reducing power. The point is that each of these individual units that get pulled off, each of these individual 2-carbon units that could get pulled off of a fatty acid, make a tremendous amount of energy. And you can imagine if your fatty acid chain is dozens of carbons long, you can make lots of energy through this process. And so fatty acid metabolism is extremely extremely awesome for generating energy. And so here you're looking at fatty acid metabolism kind of scaled out way. So here's your lipids, and then a lipase will chop those lipids into glycerol and fatty acids. Now the fatty acids, as I showed you, can go through this pathway to the beta oxidation pathway shown here to make acetyl-CoA. And the glycerol part can actually be used and shunted in to make glyceraldehyde 3-phosphate, which, and again, this is essential component of glycolysis that ultimately gets converted into 3-carbon pyruvate, and then the pyruvate can be decarboxylated to acetyl-CoA. So each of these feeds into making more acetyl-CoAs. Each of these starting materials feeds into making more acetyl-CoA, which can feed into the Krebs cycle and make you a bunch of as I show you here, reducing power and energy. And so here's some of the fates of carbon skeletons for amino acids. So lots of different kinds of amino acids can be used to make pyruvate. A few different kinds of amino acids can be used to make acetyl-CoA. And so again, lots of feedback, where amino acids can feed into this system to allow TCA cycle to generate lots of energy and lots of reducing power. And these arrows can go the opposite direction as well, depending on the state that the cell is in physiologically. Another cool pathway is the glyoxylate cycle, which is basically just a shunt reaction of TCA. And there's lots of different kinds of pathogens. Not lots. There's a few kinds of pathogens which use glyoxylate cycle. One of which is Mycobacterium tuberculosis. When this guy is growing, when these organisms are growing in the tuberculosis infected lung, they often are fed a steady diet of fatty acids. And in order to persist for long periods of time, because TB really is a chronic long-term infection, the microbes are feasting off of fatty acids for that long-term survival. And so in this particular case, the gating enzyme for normal TCA cycle is isocitrate dehydrogenase. And so in this process, the acetate, the 2-carbon acetate and 4-carbon oxaloacetate come together to make the 6-carbon citrate. Then isocitrate, it's isomerized, and then the dehydrogenase is pulled into, and you see decarboxylation, followed by production of the 5-carbon alpha-ketoglutarate, another decarboxylation to make 4-carbon succinate, and so forth. And so that's the typical TCA cycle. But the microbes like Mycobacterium tuberculosis don't want to give up all this carbon. They need that carbon for long-term survival and for the production of wax and so forth in their outer coats. Hopefully you remember doing acid-fast staining and how that relates to wax in mycobacteria. So the gating enzyme for the glyoxylate cycle is here, this isocitrate lyase. And so microbes will shut this down in favor of the shunt reaction. And again, that bypasses all this carbon loss. And so isocitrate is converted here at this gate to glyoxylate by isocitrate lyase. Actually, it makes glyoxylate and succinate. But the isocitrate lyase chops that into these two compounds. Now the glyoxylate can be mixed with a little acetate to make malate, and that can be fed backwards to pyruvate and then ultimately to make glucose through gluconeogenesis. So again, these types of reactions give microbes that have slow growth and high longevity the ability to survive for long periods of time using this kind of slow energy generating process. But it also gives us an opportunity to potentially knock these microbes out. If it's possible, because this enzyme is so unique to prokaryotes, it's possible that we could knock this thing out and limit, for example, persistence of tuberculosis. I'm not sure if anybody's really working on that. I'm sure they are, but I'm not sure if it's been successful, or else I'm sure I would have heard of it. So, lastly, just taking a look at the anaerobic food chain. So in this particular case, all these substrates, carbohydrates, amino acids, purines, pyrimidines, etc., can be broken down by fermentative type organisms to make organic acids, alcohols, and gases. And then obligate proton reducers and sulfate reducers, these types of microbes that are anaerobic, can make smaller molecules, and the same gases, taking organic acids and making simpler acetate, for example, out of those. And then methanogens can grab CO2 and acetate and hydrogen and make methane and CO2 out of that. And so again, just showing you how these big carbon molecules are progressively broken down by different groups of microbes, starting with, and this is, again, these are primarily anaerobic, but you can imagine that aerobic metabolism would be up here. Aerobic microbes like bacillus species that are breaking down huge polymers of starch and releasing individual smaller, easier to eat carbohydrates, or breaking down big polymers of proteins and leaving smaller amino acids and so forth that can then filter down into the anaerobic food chain.